Hi, Miss Nikki here. Welcome to Chapter 12, Part 1. Part 1 is a lot of definitions. So we're going to look at kind of the organization of the nervous system. We're going to talk about how nerves are classified. We're going to look at some of the unique structures that make up a neuron. And then in Part 2, we'll talk more about how an action potential is generated in a nerve. And if you've studied chapter 10 and you understand how an action potential is generated in the muscle, this will be really easy. You'll just have to learn a few different aspects, but a lot of it is, or a lot of the main ideas are repeated. So don't freak out too much by how large this chapter is. We're going to kind of cherry pick it and do a little bit more of an overview in certain areas. So let's get started. Obviously, the general function of the nervous system is going to be communication and control. So we've talked so far throughout this semester about how the brain is going to collect information and process that information and then cause some sort of action to happen in the body. Way back in chapter 1, we talked about negative feedback, right? So there was some sort of receptor that detected a stimulus and then sent that information to the spinal cord and eventually the brain. So your brain is collecting information, and the brain is part of the nervous system. And then your brain is processing that information and deciding what to do. So remember we said stimulus, receptor, control center was your brain, and then your brain sent out a message and that went to the effector, right? So the processing part is the brain. And then we have initiate, response to information. So this would be the same thing as effectors that we talked about earlier. If we're organizing the central nervous system based on structure, we're going to divide the nervous system into central nervous system, which is brain and spinal cord, and then peripheral nervous system, which is everything outside of that area. So that would be nerves, ganglia, which we'll talk a little bit more about what that means. If you remember, here's the brain, here's the spinal cord. Do you remember the intravertebral foramen? Those holes that were created between two vertebrae, well, anything past that point, that's now going to be peripheral nervous system. So that's going to include all of your spinal nerves and ganglia. If we were going to organize the nervous system according to functionality, what they do, what the nerves do, we would have sensory and motor. So sensory is carrying information to the brain. So these are afferent neurons, and they're carrying sensory information to the central nervous system. So that's brain and spinal cord. In the motor division, this is the efferent nervous system. This is where we're initiating motor outputs and transmitting them away from the central nervous system out to the effectors. This figure is showing you kind of a map or a flow of the functional organization. We can see the sensory information is traveling to the brain. So from the body, somatic means body, we can have a body message, so coming from the eyes, the ears, the skin, or we can have a visceral message, which is coming from some sort of internal organ, something that's more deep in the body. So you can have kind of an outer message or an inner message, and those sensory messages are traveling through spinal nerves to the spinal cord and going up to the brain. Remember, your brain is your control center, so it has to decide what to do with that information. And then if the brain decides that it needs to move or cause some action to happen, it's going to send a message down the spinal cord and out the spinal nerve via the motor nervous system. And then what are we going to cause to move? Does the brain initiate a somatic motor neuron? So think of skeletal muscles. Or does the brain send a message to an autonomic motor neuron? That would be anything involuntarily controlled. So you should be thinking cardiac, smooth, and any sort of gland like a sweat gland or other glands that we'll talk about in 202. So bottom line, when I hear sensory nervous system, I'm thinking of afferent, right? Message traveling to the brain, ascending. When I see motor nervous system, I'm thinking of efferent, and this is exiting the brain. And the messages can be sent from outer receptors like eye, ear, skin, or they can be sent from internal receptors. And motor messages coming out, those motor messages can go to skeletal muscle, or they can talk to or communicate with the autonomic neurons, such as the cardiac muscle or smooth muscle or any gland. So this is kind of what we've been talking about with muscle action, right? So what is a neuromuscular junction? That's when a motor neuron is talking to a somatic motor neuron, right? So it's telling skeletal muscle what to do. So here we kind of have a definition for you. Nerve is a bundle of parallel axons. You can also, or you will also hear people say a nerve is a bundle of neurons. Axon is a part of a neuron. And when we look at the parts, we'll talk about that a little bit more. But bottom line, what is a nerve? The nerve that comes out of your intervertebral foramen, it's a whole bunch of individual neurons wrapped together. And how do we wrap them? We wrap them with connective tissue. And this should look really familiar. Instead of epimysium, paramysium, endomysium, we have epineurium, perineurium, endoneurium, and each one is wrapped in connective tissue. Also remember that nerves are vascularized. So neurons are cells, so those cells have to receive nutrients. So we're going to see blood vessels innervating nerves. Nice image from your textbook for you. You can see that we have a nerve, and a nerve is a bundle of neurons, and we have epineurium wrapping around the entire nerve. Then we have fascicles, right, bundles wrapped in perineurium, and then we have the individual neuron wrapped in endoneurium. But also remember that the neuron is also going to have a plasma membrane. So all of this should be really familiar if you've studied chapter 10 and looked at the muscles already. There is some new terminology when we talk about the structure of a nerve. So we've seen a neuron before, right? Neuron has kind of a head, it has these dendrites, it has a tail, that's the axon, and then we saw the synaptic knobs, the little feet coming off of the neuron, right? So at its core, it has the nucleus. So this part is the cell body. This part is the axon, and this is the axon terminal or the axon terminal with synaptic knobs. So we saw this in chapter 10. Another word here, nerve. So you can see why they call a nerve a bundle of axons. But the nerve can also have this kind of bump or this section, which is called a ganglion. So a ganglion is a collection of cell bodies. So it's like we took a whole bunch of these heads to the neuron and we kind of put them in this area. And it really has to do with the way that the axons are running. So it's kind of like the axon runs this way, and then we have the cell body, and then the axon keeps running. So wherever you have the cell body heads, that's going to be called the ganglion. If we look at the structural classifications of nerves, we also have cranial nerves versus spinal nerves. So cranial nerves extend from the brain. Do you remember the transverse foramen? Transverse foramen in the cervical vertebrae. Remember we have the two holes, we have the vertebral foramen, and then in the transverse process we have these two extra holes. That's where the cranial nerves are going to travel or be found as they're extending from the brain to various body locations. Usually, and we're going to talk about all 12 of them later, but usually I think of cranial nerves as kind of primitive functions that we need. So think of sight. So you have your optic nerve, smell, olfactory nerve. If you hear a really loud noise and you automatically turn towards the noise, that's also controlled by a cranial nerve. So we'll talk more about those as we get through the next couple of chapters. Spinal nerves are kind of the nerves that we talk about all the time. So that's the image that we just saw with the ganglion and the nerve, right? They want you to realize that some nerves are just sensory, some nerves are just motor, and some nerves are mixed. Some are going to carry sensory information and motor information. So we'll talk again about this a little bit later, but like 2 of our cranial nerves carry sensory only. So sight and smell, cranial nerve 1 and cranial nerve number 2, olfactory. So 1 is optic and 2 is olfactory. All they're doing is sending sensory information to the brain. They have no motor function. Here's the term, or kind of the written out definition for a ganglion. So the ganglion is a cluster of neuron cell bodies in the peripheral nervous system. Neuron characteristics. 1, they're excitable. This is the only way that we can get a message from our big toe all the way to our brain, right? We have a relay race of neurons that are responding to some sort of stimulus. 2, conductivity. We can propagate an electrical signal from neuron to neuron to neuron, right? This would be the neuron action potential that we've already seen when we talked about muscles. 3, they have the ability to secrete neurotransmitters. Remember, neurotransmitters, that was the chemical messenger. So that was acetylcholine was released from the neurotransmitter in order to turn a neuron action potential into a muscle action potential. 4, they can live a very long time, 100+ years. And 5, they are amitotic. So A in front of something means not. Mitosis is to divide, right? So after fetal development in utero, they have lost the ability to copy themselves. So basically, the neurons that you're born with are all the neurons you're going to have in your lifetime. Here we have a typical neuron structure, and here is the cell body. This is kind of like the core. This is where we have the nucleus, right? DNA. We're also going to have organelles here, like mitochondria and Golgi apparatus. And out here, coming off of the cell body, are the dendrites. And the dendrites kind of receive the signal. Think of them as the receptive part of the neuron. And then from the cell body, we have this narrowing. And this narrow section has a specific name, the axon hillock. I've also seen axonal hillock, but either way is fine. So it's kind of the taper point from the cell body to where it becomes more cord-like, and this is our axon. And when we get to the end, we usually call this the axon terminal. It's the end of the axon. And then we have our synaptic knobs that we're really familiar with from the muscle chapter. I want to talk about the path that information takes through a neuron. So we can see here, this is neuron 1. And I can see my little synaptic knobs. And you can see those synaptic knobs are in close proximity to the dendrite of this neuron, which is neuron 2. So the first neuron can talk to the second neuron by releasing a neurotransmitter from the synaptic knob that can excite the neuron. It'll take an action potential or an electrical signal down the axon terminal, and cause neuron 2 to release a neurotransmitter. And what is it picked up by? It's picked up by neuron 3. So this is what we'll talk about in part 2, how you can have one neuron generating an action potential, which can trigger the second neuron to generate an action potential, release a neurotransmitter, and eventually cause action to happen on the third neuron. So the pathway is always going down. You will never see an action potential going back up. Sometimes bacteria and things can travel retrograde or back to the neuron, but when we're talking about electrical signals or action potentials, they're always going to move down from dendrite to soma or cell body to axon terminal to synaptic knob. We move on to talk about the structural classification of individual neurons. So individual neurons can take on different shapes. This is not the same as what we talked about in previous slides. We talked about the structure of nerves, so the bundles of neurons. And we said that we had cranial and we had spinal, right? Those were the structural classifications of nerves. Now we're looking at the structural classification of individual neurons. So they can be multipolar. This is the most common. This is what we've seen. So we have a cell body, we have dendrites, we have an axon hillock, we have an axon terminal, and we have our little feet or synaptic knobs, right? So that's the one we've been looking at. Next, we have bipolar neurons. You're going to have a few dendrites at the top. Really, technically, this is the dendrite. But you can kind of have this fan at the top, then dendrite, then soma, then one axon. And then you're still going to have the ability to release neurotransmitters through the synaptic knobs. So bipolar is kind of consolidating information, collecting information. And this happens in the retina of the eye. So we're kind of taking a whole bunch of information. We're taking a whole bunch of information here, and we're consolidating it on this dendrite to send a quick signal down the axon, release a neurotransmitter, and have some sort of action happen. Unipolar I think of as very quick. So we'll see these in reflexes, where unipolar, you're going to have the dendrite, and the soma is going to be in a different position where you can go from the dendrite directly to the axon without having to go through the cell body. So it allows for a quicker electrical current or a quicker action potential being spread where you don't have to waste time going through the cell body. And then we have these neurons that have no axons. So anaxonic. This means not, right? So no axons. And the purpose of this would be to kind of gather information. So you have a cell body and you have nothing but dendrites. And remember, dendrites are the receptive end. They're receiving messages. So we're going to see these types of neurons in what we would call interneurons in the brain, where they're just collecting information. Here we have the functional class of individual neurons. So they're either sensory, motor, or interneurons. So again, we're going to have the same setup where we have sensory information, right, going to an interneuron, and then that interneuron sending out a motor message. There's a lot to kind of unpack with this image, but let's kind of do an overview. We have skin receptors. There has to be some sort of stimulus to excite the receptor to cause some sort of electrical change, right? So we're generating or exciting this neuron, and the message is traveling through the neuron, and the neuron is in a bundle which is called a nerve. We can see the cell body here. After the cell body, what does it become? It's an axon, right? And then what's at the end of the axon? We have synaptic knobs. Synaptic knobs are going to release neurotransmitters, and basically it's a way for this sensory neuron to talk to this interneuron. So the neurotransmitter is going to be released, it's going to bind to a receptor, it's going to generate a potential in the interneuron, which is then going to release a neurotransmitter and generate a potential in a motor neuron. I want to make sure we understand that this is just one way, one pathway. What else can happen? You can have a sensory nerve generating a potential. It can get to this point of the spinal cord, and we can send that information up to the brain. The brain can also have interneurons that are going to process information. Then the brain would send a message back down to the motor neuron and cause action to happen in the body. So you can have interneurons in the spinal cord or in the brain. Synapse is the term that we use to talk about one neuron communicating with another neuron. Chemical synapse is what we're familiar with, right? Way more common. Chemical synapse is where we release neurotransmitters. Neuron 1 talks to neuron 2 via a neurotransmitter. Electrical synapse, this is mostly in the brain where we need really fast signals. So there's neuron 1 talking to neuron 2, but they're bound together. So there is no synaptic cleft, if you remember the synaptic cleft, the space between the two neurons. So here's my neuron 1, here's my neuron 2. This space would be called a synapse, where they're talking to each other. And in a chemical that we saw, we'd see a neurotransmitter released into the synaptic cleft. So this would be an example of a chemical synapse. Electrical synapse would be gap junctions actually connecting two membranes so that the neurons are actually bound together. There is no space between them. More detailed kind of analysis of chemical synapse, they just want you to realize that if you have one neuron, it's going to release a neurotransmitter to talk to neuron number 2. And if this is called a synapse, two neurons talking to each other, this one is going to be the presynaptic neuron, and this one is going to be the postsynaptic neuron. And because we're releasing a neurotransmitter, and because it has to be picked up by the dendrite of the second neuron, we have to generate a potential. That potential has to travel down the axon and cause action to happen at the synaptic knob of the second neuron. This takes time, right? There's a delay in time for all these events to occur. So electrical synapse is fast, usually found in the brain. Chemical synapse is what we're most familiar with, where we release a neurotransmitter, but it is going to cost a little bit more time. If we were to look at nervous tissue sample, we can see here the cell body. We can see the dendrites coming off of the cell body. We can see the taper or the axon hillock. We can see the axon. You see these cells kind of surrounding the neurons? Those are the glial cells. These are supporting cells. They have nothing to do with generating an electrical potential. And we find them in both the central nervous system and the peripheral nervous system. These cells, these helpers, are capable of mitosis. So remember, neurons are not. They're amitotic. And they're really going to help kind of protect and nourish neurons. They're going to provide some sort of scaffolding or stability in the nervous tissue. And they're critical for normal function at neural synapses. We have 4 glial cells that are found only in the central nervous system. The first is an astrocyte. This helps form the blood-brain barrier. So you can see the astrocyte here with its cute little perivascular feet wrapped around the blood vessel. Or capillary is just the smallest blood vessel, right? So basically, I think of these as the gate guard. What's allowed to go from the blood to the brain? Obviously, we wouldn't want toxins or pathogens to get to our brain. That's our supercomputer, right? Ependymal cells are going to line the brain and the spinal cord, and they're going to help produce cerebral spinal fluid. Look at the structure of the ependymal cells. Simple, right? Cuboidal. What does epithelium do? Especially if I'm simple epithelium, right? Either absorb or secrete. So these ependymal cells, I can tell just by looking at them that they're going to have some absorption or secretion function, and that way we can easily relate it to helping produce cerebral spinal fluid, which is going to help kind of wash the brain. Next, we have microglial cells. Probably the best way to think of these is phagocytic cells. So kind of like macrophages, they're going to engulf any pathogens that are not supposed to be there. They're going to do the endocytosis, use the digestive chemicals to break down this cell or pathogen or anything that's not supposed to be there. And then lastly, we have oligodendrocytes. Oligodendrocytes are these kind of cyclops-looking cells, and they reach out and they wrap the axons of neurons with myelin. So it says myelinates and insulates central nervous system axons. So we'll talk a little bit more about myelin, but think about, in cold weather when you wrap pipes, right? You're insulating. In this case, we're trying to create a faster signal. We're trying to allow for kind of a quick signal so that nothing is kind of escaping. So if you wrap myelin or you insulate the axon, you'll have a faster signal conduction. Next, we have two glial cells that are only in the peripheral nervous system, so outside of the brain and the spinal cord. The first one is called a satellite cell, and you can see here in pink that the satellite cell kind of insulates or wraps around the cell bodies that are in the ganglion of the peripheral nervous system, and it helps kind of regulate nutrients and waste exchange. They used to say these were similar to astrocytes, and for a while we didn't know a lot about them, so they're kind of ongoing in their research. The second type of glial cell we have in the peripheral nervous system is called a neuroleukocyte. And this is going to be hard for me because I want to call it a Schwann cell. And you might see this still on your list as a Schwann cell. You might see it in APR. This textbook likes this term, neuroleukocyte. And one of the things that I don't know if I've mentioned, they're trying to get away from eponyms. So eponyms are when you name a structure after the person who found it, right? It's naming. And they're trying to get to more of its function with its name. So neuroleukocytes also produce myelin, and they insulate or wrap around the axons of the peripheral nervous system. So you can see this is very similar to an oligodendrocyte, right? This is a nice image of the neuroleukocyte kind of wrapping. So here is the cell, the neuroleukocyte. I know it's a cell because it says cyte, right? And it has a nucleus, and this neuroleukocyte is going to create a protein, and it's a high fat, so it's kind of a lipid protein. And that lipid protein is what makes up myelin. So the neuroleukocyte is just making myelin, and it's wrapping at the same time. So eventually you end up with this myelin sheath, this wrapping of myelin around the axon. One of the things to notice when we look at a neuroleukocyte, we can see that these are individual cells, so each one has a nucleus. If we contrast this to oligodendrocytes in the central nervous system, you can see the majority of the cell here, and you can see the oligodendrocyte is reaching out and wrapping several different axons. So multiple axons on different neurons can all be wrapped in myelin by one oligodendrocyte. Here we have two disorders that affect myelin. We have multiple sclerosis, so this is autoimmune disorder, so we should be thinking self-attacking, right? So our immune system is attacking the oligodendrocytes that are in the central nervous system, and that's causing demyelination of the neurons. You're going to have repeated inflammatory events, there's going to be scarring, and there can be permanent damage and loss of function. In Guillain-Barré syndrome, this is kind of a temporary, so you can have some recovery with little medical intervention. So there is a loss of myelin due to some sort of inflammatory event. It can cause temporary muscle weaknesses and advance to proximal muscles, which would not be great, right, as we move towards the trunk with breathing, the intercostals, the diaphragm, right? So the myelin can be affected. And that wouldn't affect necessarily the nerve, it would just affect the nerve function. In the peripheral nervous system, we have seen axons regenerate. If the cell body is intact, it's possible to have regeneration. Central nervous system, we do see some regeneration, but it's extremely limited. There's usually going to be some sort of scarring in the connective tissue, and it's going to obstruct growth. So central nervous system, brain, spinal cord, very limited regeneration. Peripheral nervous system, it just depends on the extent of the damage. Now we get to move on and talk about pumps and channels within the neuron. So we're going to talk about how action potentials are generated, so they want to do a little prep work and make sure that we remember how pumps work and how channels work. So remember when we talked about the sodium-potassium pump, we said that it required ATP energy. It's a form of active transport. So hopefully we remember that. But there are also calcium pumps in neurons. There are also channels in a neuron membrane, two that we've seen before. So we have our chemical gated channel. They're normally closed but open when a neurotransmitter binds. So you remember we had the acetylcholine receptor, And what bound to that? Acetylcholine. And then what happened to the channel? It opened. So that's a chemically gated channel. For a voltage gated channel, they're normally closed, but they open when the charge changes at the membrane. So this would be the sodium only channel or the potassium only channel that we saw with muscle contraction. There is another channel listed here. There are some leaky channels. They're always open and they just kind of have a continuous diffusion of ions. Voltage-gated sodium channels have two different gates that they can close. So we have this little swing arm, right, that we can close, and then they can also close this inner section. So because these channels have two gates, they can be in different states. So they can be in a resting state where the activation gate is closed. Then they can be in an activated state where the activation gate is open and so is the internal. They call it the inactivation gate. I know it's kind of confusing, right? So both, think of this as both gates open. And what do we know? If both gates are open, what's happening to the sodium? It's going from the outside in the interstitial fluid here to the inside cytosol of the neuron. Remember, we're talking about neurons now. And then inactivation, we have the inactivation gate closed, but we still have this little foot gate, the activation gate, still open. But in a sense, you still can't get sodium in. So both of these, no sodium is making it inside, and this one, no sodium is making it inside. Now talking about channels, we can see here that we've got a sodium-potassium channel. We're going to have a leaky sodium and a leaky potassium channel. So these, the ions are just flowing based on diffusion, high to low. The sodium-potassium pump, this is active transport against the current, right? High to low requires ATP. See why we had to learn all this stuff way back in the beginning? All of this is going to help set up the resting membrane potential of the neuron. Very nice image here, color-coded for you, talking about the different segments of the neuron. And this will make a little bit more sense when we go through how we generate an action potential. And you'll see why we want to label things receptive segment, an initial segment, and conductive segment. But you can see here the receptive part, this is going to be the soma and the dendrites. Or soma, you can also say cell body, right? I learned soma, so sometimes it's hard to break old habits, right? Then here in the axon hillock, this is where you're going to have the voltage gates. So does this kind of look familiar where in the receptive segment we have chemical, right? Some sort of neurotransmitter has to bind. And then here in the initial segment, this is where we have our voltage only gates. That should look really similar to what we talked about with muscle, right? And then the conductive segment, it's also going to have voltage-gated channels. So as the inside of the neuron becomes positive, it's going to open these channels and allow more sodium ions to flow into the axon. And that's going to keep conducting all the way down to the end where the synaptic knobs are located. When we finally get to the synaptic knob, we're going to have this transmissive segment. And this is where we're going to see calcium pumps and the voltage-gated calcium channel. So remember we had our synaptic knob and it had a calcium channel. And what caused that calcium channel to open was when you had an action potential travel the length of the axon, reach the synaptic knob, and then this calcium gate opened. And when the calcium gate opens, we're going to see a flood of calcium enter the synaptic knob, and that's enough to trigger the vesicles to release their neurotransmitter. But remember, we also have to have a calcium pump so that we can pump that calcium back outside of the neuron for the next time that that synaptic knob needs to release its neurotransmitters. This is a beautiful figure to kind of give you an idea of what's going on around the neuron and inside the neuron. So first, let's look at ions. So what do you notice about this section, right? The cell body and the dendrites. You can see that there are more sodiums and more chlorides. So we have increased sodium, increased chloride ions. And then on the synaptic knob or transmissive segment, you can see that we have a lot more calcium ions. Inside, we have more potassium ions. So potassium, this is inside. These are all outside. Hopefully we remember interstitial fluid. Another thing to notice, negative 70 millivolts is the resting membrane potential inside the neuron. Remember, these chemical gates will be closed during resting membrane potential. What's going to happen? Eventually a neurotransmitter is going to cause them to open, right? And then we're going to see the inside become positive, and we're going to see that message spread down the axon. And remember, the axon has the sodium and the potassium only voltage gates. So that's what helps propagate the message down the axon. I wanted to show you that the leaky channels here, both sodium and potassium, lead to kind of setting up this resting membrane potential at negative 70. So the potassium is going to leak out, right? It's going from high to low through diffusion. And then we have sodium leaky channels. This is high to low. And while these leaky channels are letting ions in or out, we also have here the sodium-potassium pumps that are kind of setting the concentration gradient, right? Setting the amount of sodium and potassium. Probably the most important one is the potassium leaky channel. If we didn't have the sodium, we'd be at about negative 90 millivolts, because as more positive potassiums leave, remember those really large negative phosphate molecules would make the inside even more negative. So we kind of have a balance going on here. Here's an overview of the events of each neuron segment. What they're saying is there's going to be some sort of stimulus here. There has to be a receptor, right? Some sort of chemical voltage gate, right? So there's going to be a stimulus. That stimulus is going to cause the receptor to open. That's going to cause a charge difference inside the soma or the dendrite. And when it gets to the axon hillock, remember that's where the sodium only gate was. Once the potential or the charge difference reaches this axon hillock, these sodium only gates are going to open, and it's going to cause a rush of positive sodium ions. And then it's going to keep opening sodium only gates so that that message is propagated or conducted down to the synaptic knobs. And when we get to the synaptic knobs, remember we have those calcium voltage gates. And they're going to open and allow the calcium to enter the synaptic knob, which will then cause the vesicle to drop its neurotransmitter. So basically we're talking an overview of how do we go from step 1, stimulating a neuron, to the very end where we have the neurotransmitter released.